the mechanobiology part. You can tell, been here listening to these talks, and based on what we've heard, I sort of modified what I'm talking about, and I have three missions, I think, today. The one is I'm going to take some of the terminology that you heard before as words, integrins, adherins, and I'm going to tell you a little bit more about them so you can understand some of the underlying principles. Second, Ben Prosser used this term, mechanotransduction. How do you convert a mechanical signal into a chemical signal? Mike Ostap talked a lot, or some about that. I want to talk more about the chemistry and how do you actually go from a force to a chemical pathway, and I'm going to give you some examples of that. And I only want you to remember the concepts, not any of the detailed information. And the third thing we've heard a lot about this week is that cells use actin to regulate tension in the cell, but we haven't talked about how do you regulate that. And I'm going to talk about a family of proteins called row family GTPases that you can't live in mechanobiology without hearing about. So I'm going to introduce those to you. So those are my three missions. So let me talk a little about integrins. So we've heard this word a lot in this week. So integrins, we've heard, are plasma membrane receptors for the extracellular matrix proteins like collagen and fibronectin and laminins, and we've heard that they connect to the actin cytoskeleton. This is what a model of integrins look like. The sort of the conceptual parts that you want to think about, very large. It's a heterodimer, alpha chain and a beta chain, and that's going to become important later. Very large extracellular domain, very tiny intracellular domain. And one of the standout features of the integrins and also in adherins, which is different from many, many receptors, is that there is no enzymatic activity in this intracellular domain. Now you hear about receptor tyrosine kinases, right? You bind the ligand, they turn on a kinase as part of the receptor. These intracellular domains are much too small, 20 to 40 amino acids, to have enzymatic activity. Yet they have to transduce information. Mike Ostap talked about the activation of integrins, and I just want to bring up, one or two small things. We talked about the inactive integrin being in this floppy form. This is the active integrin in this straightened up form. The important part for mechanotransduction, going from force to chemistry, is down here. In the inactive low affinity form, these cytoplasmic tails are in proximity to each other. When the integrin is opened with force, these tails separate. So the alpha and the beta subunits now have exposed tails. And that's really important because, hold on, this region of the exposed tail is the binding site for tailin. And so when you pull on the integrin, you expose a binding site, tailin can bind, and then you can start transducing that force into the cell, as you've heard about before. So why do we have integrin heterodimers? Integrin heterodimers are critical because that's how you determine ligand specificity. So if you look at the ligand, so here are collagen receptors. This is a little bit fuzzy, but they're alpha 1, alpha 2, alpha 10, alpha 11. Don't remember the numbers at all. There are alpha subunits that all heterodimerize with a common beta subunit, and these form collagen receptors. There are other alpha subunits, 5B, all of these, that heterodimerize with that same beta 1 subunit, but these form fibronectin or RGD receptors. Because so the alpha subunit will commonly change and use a common beta 1, that will let the cells bind different ligands. Okay, sometimes the beta subunit changes, many times the alpha subunit changes. And so this ligand specificity in the integrin family is due to these different combinations of heterodimers. We've heard about from I can't remember if it was Mike or Ben, inside and outside signaling. This is also a term used in the integrin field, where inside-out signaling relates to the ways that cells put tension on integrins to activate them, the way I explained in that last diagram. And outside-in signaling is how the ligand binding to the receptor transduces a signaling pathway that leads to cell decisions. Okay, and we're going to talk some more about this soon. We've heard a lot about the term focal adhesion, so I want to talk a little bit more about that terminology. We all, you've heard that focal adhesions contain integrins, and it's how the ECM is connected to actin. So you may hear other terms as you go through your time in mechanobiology: focal contacts, focal complexes, focal adhesions. These are different terms that they use to represent the maturation of this complex from relatively simple complexes in size and composition to these more complex, these yellow things are the focal adhesions that we know and love. But there's increasing recruitment of components. There's this focal contact, this very initial adhesion is force independent. And as these components get added to the focal adhesion, then those are force-dependent processes. And you'll hear about this term maturation of focal adhesions. It's going from these early focal contacts through these complexes into the mature adhesion with force. Here is the, this is a very outdated view of the complexity of the focal adhesion. And I just want to show this as an example that all of these things are kinases or adapters, right? So that we've heard about the idea that focal adhesions transduce the physical information into the chemical information. But I want to talk about a little more detail about how, okay? Because ultimately, mechanobiology, or a large part of mechanobiology, is chemistry. And here is here's one of the really important concepts which spans just all of biochemistry and cell biology, well beyond mechanobiology, okay? Proteins are made of motifs. I don't think this is projecting very well, but there's a motif structure. There are common sequences in proteins, and I've listed some of them here. The people who are on the biology side have probably heard these already: SH2, PTB, SH3, WW. These are domain structures that have the ability to bind other motifs. And I'll give you a couple. So an SH2 domain protein, to a protein that has an SH2 domain, this is an example of SARC. There's an SH3 domain, SH2 domain, and a kinase domain. SARC, SH2 domain, will bind the phosphotyrosine in another protein. And that's sort of the chemical basis for how proteins get recruited to other proteins and how you assemble these focal adhesions through the binding of proteins to each other through these different motifs. So a pH domain binds phospholipids. It's the way proteins get recruited to the inner plasma membrane. The SH3 domain binds proline-rich sequences. And I'm going to give you a couple examples of this chemistry at play in the mechanobiology context. One of the enzymes we have not heard said here, but is very well studied mechanobiology enzyme is focal adhesion kinase, or also called FAK. And it is a classic way that integrins transduce information. And so here's a cartoon diagram of FAK, and there are several tyrosines. FAK has a kinase domain at its C-terminus, and and it has what's called this autophosphorylation site. There's a tyrosine here. It's got a bunch of other tyrosines, okay? What happens when integrins get clustered, the associated FAK, so FAKs bind to integrins. These integrins come close to each other, and one integrin will phosphorylate the other, and the other integrin will phosphorylate back. And what you get what's called transphosphorylation or autophosphorylation at this site, okay? So now you go from a tyrosine to a phosphotyrosine, now you form an SH2 domain, and that SH2 domain, it's a non-covalent chemical interaction, and you change the affinity of that site for SARC. Because you phosphorylated that tyrosine, now the SH2 domain on SARC has a good enough affinity that it can bind to that phosphotyrosine, and so you recruit SARC. It's an example how proteins are recruited to other proteins through these non-covalent interactions. Then SARC itself is a tyrosine kinase, and now it's in proximity to all of these other tyrosines on fat, and it starts to phosphorylate these other tyrosines. This tyrosine enhances the kinase activity of fat itself. These other tyrosines then become SH2 site domains, then become phosphotyrosines that recruit SH2 domains of other proteins, and now you can sort of see how you build the complexity of the focal adhesion through these non-covalent chemical interactions. Okay, that's what I said there. This is two examples of how you see mechanosensing of fat. These are cells that are stretched at different times, and what you see in this is a Western blot of cell lysates, that the total amount of fat in these different stretched cells is constant, but the phosphorylation at the autophosphorylation site increases. Here's an example of cells that were placed on soft or stiff hydrogels. If you look at total fat, it stays relatively constant here, soft, stiff, soft, stiff, soft, stiff. But these different tyrosine phosphorylation sites increase, as does phosphorylation of other focal adhesion proteins. Again, using this, this common paradigm of protein motifs, phosphorylation, and changing in the affinity of these non-covalent interactions. Mike Ostap talked about a really critical concept. I'm saying again because it's fundamental explanation for molecular mechanobiology, is that force pulls domains apart or can push things together. And and that can change chemistry. So here's an example in Cas. Mike used a different example where Cas, P130 Cas is anchored at its N and C terminus and pulled apart, and that reveals a tyrosine in the coiled area here, and that tyrosine can then become phosphorylated with force. Why do we care about that? Because it now allows SH2 proteins to bind to Cas, and you perpetuate the buildup of focal adhesion components. Did you have a question?
Yeah. So in the previous slide where you showed increasing correlation.
Yeah.
The fact that remains constant.
Yes.
Is that increase due to increased number of focal adhesions or increased clustering of the integrins that are already there?
And this paper did not look at it at that level of detail, but either if you did the experiment kinetically, you should be able to address that. Either of those explanations could give you more phosphofat. So, focal adhesion, part of the complexity is you use force to build up focal adhesions, but then once focal adhesions are formed, they're self-reinforcing and increase force, right? So either of your explanations would be fine. You heard about, from Ben Prosser, mentioned very briefly this thing called YAP-TAZ. I want to talk about YAP-TAZ just a little bit more, and then another mechanosensitive transcription transcriptional regulator called MRTF, myocardium-related transcription factors. I point out these two because these two transcription factors are the paradigms for how you transduce information from the cytoplasm to the nucleus under force. The YAP is actually not a transcription factor. It's a detail at this level, but it regulates the activity of transcription factor. So it has to bind to a transcription factor in the nucleus. So YAP, as Ben mentioned, is in the cytoplasm. And when cells are under tension, it transduces and binds to its recognized transcription factor partner called TEAD. And the binding of YAP to TEAD allows T to regulate transcriptional output. The better studied one is actually, at a chemical level, is actually MRTF, which has several other names, but we'll call it MRTF. MRTF, like YAP, is retained in the cytoplasm, and under force is transduced to the nucleus, where it is a bona fide transcription factor. But we understand much better how MRTF is regulated at a chemical level. So here's MRTF binding to G-actin, is the monomer. This little arrowhead is the monomer, and here are the polymers of that monomer built into F-actin. When G-actin gets incorporated into F-actin, MRTF is dissociated, but what this binding event does is it blocks what's called a nuclear localization sequence that's in MRTF. It's a peptide sequence that lets proteins enter the nucleus. So that sequence is hidden in this G-actin complex. When the G-actin is taken away, now the nuclear localization sequence is exposed, and the escort proteins that can bind to that sequence, again, non-chemical, exposure or change in the affinity of non-covalent interactions, and carry MRTF into the nucleus. You will hear a lot about YAP and MRTFs. Okay, we also heard the words coherins and catenins, and I want to talk a little bit about that. So this is a diagram of a cell-cell junction. These are also used a terminology of both adherens junctions. It's sort of the counterpart of focal adhesions. So these are two cells, each of which have an E-coherin in this molecule sticking out. These form non-covalent, again, interactions, homophilic interactions, meaning that one molecule binds another molecule. Okay, and that is the basis of a coherent interaction. There is a conceptual counterpart to, there is a conceptual counterpart to the focal adhesion at an adherens junction, in the sense that you see this buildup of non-covalent interactions of signaling molecules in conceptually the same way that they're built up at a focal adhesion. Some of these molecules are the same. There's vinculin in both of these. Some of these molecules are different. So here, again, this is not projecting very well. There's alpha and beta catenins and P120 catenins in the adherens junction that you don't have in the focal adhesion. And why is that important? And that's important because the catenins are how coherins attach to actin. And the way this works is, look just right over here. So the intracellular tail of a coherin, just like integrins, is very small and does not have enzymatic activity. It has to work by recruiting things with activity to it. Okay? It recruits a molecule called beta-catenin. Beta-catenin, which is this green molecule, then binds alpha-catenin, and alpha-catenin has an actin binding site. How does alpha-catenin bind actin? The exact same way that Mike talked about. There are forces that open the structure of actin, reveal sites that can now bind to the actin filament. So what I wanted, the really interesting thing about a coherin-coherin interaction is shown here, is that, hold on, let me go back to this one a second. So we're coordinating this junction with actin. We're coordinating the extracellular matrix with actin. And so there's crosstalk in a mechanical way between this cell-cell adhesion system and this one. So that means that if there's force applied on this cell, that force goes through the cell-cell junction and can be detected in this cell. So if this cell over here on the right starts moving towards this wall, because of this junction and the way these forces can be now interconnected between cells, these cells can now feel that force that was driving the migration initially over here. That's the basis for a term called collective migration, where cells move as units as opposed to individually. Okay, and I want to end in the last part about how is tension regulated, okay, inside a cell. There is a family of molecules called Rho family GTPases. So let me just back up a second. There are two very different kinds of chemical modifications that regulate signaling that you don't remember very much. Remember, proteins have different motifs that change their affinities, and that there are different kinds, these two different kinds of signaling interactions. One are covalent interactions. Phosphorylation of a protein is a covalent modification regulated by kinases. There is another signaling group that are not kinases or not phosphatases, which remove that phosphate, but are called GTPases. These are non-covalent activations. So the GTP, so kinases use ATP to regulate phosphorylation. Rho family GTPs use GTP. And when a GTPase binds GTP, it's in an active form, it can transduce information. When that GTP gets hydrolyzed back to the GDP, it is in an inactive form. And there are 3 Rho family GTPases that regulate the cytoskeleton in the cell. One are the Rho family, which you probably have heard about the most, and we'll talk a little bit about that. That regulates stress fibers. You've seen many people talk about stress fibers here. And RAC GTP regulates a morphology called lamellipodia. I'll talk a little bit about that. There was a picture that passed by of beautiful filopodia. I don't think the word was said previously, but another GTPase, CDC42, regulates filopodia. I'll show you a picture with each of these kinds of things. And these are all regulating, through this non-covalent binding of GTP to the Rho, RAC, or CDC42. So here are some pictures from a classic paper from Alan Hall's lab where they first identified these GTPases. Here is a basal fibroblast. If you put in an active form of Rho, you see all of these beautiful stress fibers that we've seen lots of pictures of, okay? If you put in an active form of RAC, you don't get stress fibers at all. You get these things, what's called ruffling. I'm going to show you a much better picture in a minute. This sort of like the edges of the cell are folding up. That's what you're seeing in this still diagram. And if you put in CDC42, you get another unique morphology called filopodia. Think about F filopodia, F fingers. You get these finger-like projections out of the cell. Here's a movie from my lab, which is not biologically very exciting, but shows very nice images. These are lamellipodia. You can see these blurbs fall leading this cell. Lamellipodia lead the direction of cell migration. These are cells where their actin has been intrinsically labeled. And you see the stress fibers here in this cell, and then which is Rho regulated process. And then you see RAC activation as what's called the leading edge as these things, lamellipodia shoot out. There's a lot of interest in the forms of actin that regulate stress fibers versus lamellipodia, and what's called cortical actin versus stress fibers. And so there's a lot of chemistry that's known about how do you regulate Rho family GTPases. I'm just showing you Rho, and I don't want you to know any of these details, other than to say that how does Rho regulate actin dynamics? Rho regulates an enzyme called Rho kinase, and Rho kinase has 2 important kinds of effects. One, it regulates actin polymerization through this arm, and another, it regulates contractility, and Mike talked about this through myosin light chain kinase, regulation of the phosphorylation of myosin light chain. You're going to hear, you can't get through mechanobiology without hearing about 2 inhibitors, Y27632 and blebistatin. A lot of people use these two inhibitors interchangeably. The lesson from this scheme is that these things are not the same. Y27632 is going to inhibit Rho kinase and is going to affect actin polymerization and contractility. Levastatin is a specific regulator of contractility. You'll see papers that misuse Y27632 all the time as a specific inhibitor of contractility. And so I want to, and this is my last slide. Oh, I'm very disappointed in you. I didn't hear any groans about the play on words that I put up there. Thank you. That's what I was expecting to hear. So what you want to remember, so there are adhesion receptors, the integrins combine in their heterodimers to interact and recognize different components of the ECM. And those nucleate actin. And adherins communicate within cells, and those also recruit actin. But that these two pools of actin crosstalk with each other, and that they are regulated by Rho family GTPases, both in stress fibers and at the cortex. And so if you are affecting tension over here, you can affect things going through actin all the way over here and vice versa. So there's this tug back and forth between these two adhesion systems, and you have to view these kind of things in context. With the Rho family GTPasers sort of as the regulator sitting on top of that. So if you, for inside-out signaling, you activate Rho, you're going to activate integrins. For outside-in signaling, integrins regulate Rho activity, and they regulate this balance between cell-cell and cell ECM adhesion, and through the tensional state of the cell and this crosstalk in actin. Okay, and with that, I am going to stop. Any questions? Yeah.
Whenever we talk about transduction, we always think about force being transmitted to a signaling cascade, right?
Right.
There's some sort of like expression. I wonder if you know whether there's work a lot of research out there on this topic, but how much is known about force actually affecting gene expression directly
Without a chemical step.
Exactly.
So what's, so mechanics without the transduction part. There is a little bit known, but it's not very well known. You can ask two questions. Conceptually, how could it work? And second, how important is it? I mean, how many examples are there of that happening? I don't know whether you guys know about it. I don't know of any. I can answer the conceptual one. It's conceptual that there's data that if you pull on forces, because you remember Ben Prosser talked about the Sun link complex and how that links the actin cytoskeleton and the cytosol to the nucleus and their nuclear pore complexes, which are the whole, the way things get in and out of the nucleus, right? So there are papers that show if you exert force on cells that you can, as your cell stretches, you can open a nuclear pore complex. So you can conceive of a transcription factor that could therefore just go right into the nucleus without any chemistry, right? But of the two best known studies, two best known examples, which are YAP as a coactivator and MRTF, that's not the way they work. So they use that chemistry in between. So I'm not aware of a strong example. Do you have it?
Well, so I don't know a good example. That was actually my stuff. You didn't find the answer, so I don't know that. We put together a review about a decade ago now that shows not only to the nuclear pores, but you actually connect all the way mechanically from the extracellular matrix to the integrins through the actin cytoskeleton all the way to the nucleus. And then the nucleus is actually a stiffer but also very connected mechanical system as well with all the lamins and all the protein inside. So there is a hypothesis that you could actually not only just let things into the nucleus your core, but actually distort some of the heterochromatin or some way open up some direct signaling that way. It's been a difficult problem to address, but I thought I saw there's some interesting work from Inwan's lab, I thought, recently, but I don't know that they got directly to the.
Yeah, I don't know of a good, I mean, a real clear example where chemistry is not involved, but conceptually, there's the idea floating out there that it may not need to be involved, but that doesn't mean it happens.
I think that if rearranging the chromatin organization could change the landscape, and that would be.
Yeah, so you might not even have to go through the pore complex. As you said, you could just rearrange the chromatin structure and, you know, reveal a promoter or something like that, right?
So the hypothesis is definitely there. I don't think it's going to be proven in any way.
Yeah. Okay, I think because we should take a break now because there's the next talk is in 10 minutes.